put that spike protein gene DNA inside the adenovirus. This gets inside your cell. It puts that spike protein gene inside your cell, produces RNA that's here to make spike proteins over here. So I'm going through very quickly because I think Dr. Corin's going to spend a little bit of time. But when we look at the vaccines, what did we look at when we were on there? One of the things we were focused on, especially in the EMA, was did they really look at the populations that are in the United States? And as you can see, for the Moderna, this is only after enrolling 30,000. The United States population had about 42.2% Black African American, and overall 10.2. They had some outside of the United States. So we felt that was a significant diversity within the population. Would like to see more, but if you look at other studies where they had 2, 3, 4% or less, this is a great improvement. Same thing with the Pfizer BioNTech COVID. This is a breakdown. At this time, they eventually had over 44,000. At this point, they had 37,000, but they had approximately 9.2% Black African Americans. But you can see here in the Pfizer BioNTech, they also had small numbers from other countries. Other countries included Argentina, Brazil, South Africa, and Germany, I think. And this is just the amounts they had from South Africa and Brazil, assuming that most of these were probably Black. So both of them had 10% or more participation of Blacks in the trials. If we're talking about subgroups, meaning how did the efficacy and safety work, we're talking about age, sex, ethnicity. As you can see, very good overall for the Moderna, 94.1. For the Pfizer vaccine, it was greater than 95. And even when you look at communities of color, the effective rate was very high. So in summary, both the Pfizer and Moderna vaccines, phase 3 trials, both of them, messenger RNA vaccines, found to have greater than 94% efficacy preventing coronavirus disease compared to placebo. Efficacy and safety were observed to be consistent across age, gender, race, ethnicity, as well as adults over 65. These vaccines were approved by the FDA, CDC groups for emergency use authorization. Both the National Medical Association, COVID Task Force, and their scientists reviewed this data, as well as the California Scientific Review Group, and came to independent conclusions that they agreed with this recommendation. What is the difference? And now that it's approved, how do you get it out there? And there is significant differences in the storage and distribution abilities of both these vaccines. I'm sure you heard that the Pfizer is a 2-dose, 3 weeks apart. The issue is it has to be stored in severe deep temperatures, -70. And then once when it's shipped, and you can take it out, put it in the refrigerator at 2 to 8 degrees centigrade. But once when you put it in the refrigerator, it must be used within 5 days. After mixing it, you need to use it in 6 hours. All of this can cause issues, especially because it's shipped in 1,000-dose containers. And once when you start breaking up those containers, so you need to have storage capacity and the ability to implement vaccination very, very quickly. As opposed to the Moderna vaccine, why is the messenger RNA Moderna a little bit more stable? I believe I heard because it has to do with the lipid particles a little bit thicker, but whatever it is, it's shipped, still needs to be frozen, but not as deep freeze. It's shipped in cartons of only 100 doses. Once when it gets to the place, you can actually put it in your freezer. Once when you take it out of the freezer, you just put it in the refrigerator. It's good for 30 days. And then at room temperature, you got to use it within 12 hours. It comes in multiple dose vials. Once when you reconstitute it, you must use it in 6 hours. So these are the limitations of storage and administration. So we talk about vaccine. How do we get it to the places where it needs to be gotten to in an equitable way? Here in California, we have adopted the ACIP plan to distribute this vaccine in phases. Phase 1, phase 2, and phase 3. As you can see, phase 1 has phase 1A, 1B, and 1C. Phase 1 is where we are now. This is at the recommendation of ACIP, healthcare personnel and long-term care facility residents. So you say, why only this? Well, it really has to do with availability. It was clear that when these vaccines were approved, they would not have enough to give to everybody all at once, so they had to prioritize. So this was adopted here in California. Phase 1B is going to be essential workers, educators, agriculture, police, firefighters, etc. Then we get to phase 1C, adults with high-risk medical conditions, adults greater than 65, and then phase 2 and 3, everyone else. How quickly we get to these phases is really about the distribution process, ability, and amount of vaccine that you have. We in this country do not have the strongest public health system. It's very fragmented. It's not even fragmented within states. And I think one of the reasons why we're slow at doing this is because of that fact. So within phase 1A, that includes healthcare personnel, that includes employees and residents of long-term care facilities, is tier. So even in 1A, there's tier 1, tier 2, and tier 3. I'm not going to spend much time here, but for tier 1, we're talking about high priority acute care, psychiatric, acute care medical and acute care psychiatric hospitals, and skilled nursing care facilities, including, and then we go down here to paramedics, dialysis centers, etc. Tier 2 now talks about primary care physicians. Now, I've been hearing all over the state that my friends are getting the vaccine. I'm still seeing patients, telemedicine in the office. I haven't gotten my vaccine yet, so I'm going to have to go back and figure that out. Hopefully, I will be getting mine soon. When we get to the general population, what is the plan to administer this here in California? And I can tell you on a statewide level, in Eastern California, they're adopting what they call the Health Equity Matrix based upon Healthy Places Index. What is this? Healthy Places Index is a community-based, data-driven process in which you take 8 different social determinants of health, and you give it a number. So what is the economic status of a geographic area by zip code or census tract? What is the educational level, transportation issues, social issues, neighborhood environment, clean environment, housing, healthcare. So all of these have a number. And the healthier the community, the greater the number. I don't know if you can see this, but really what I want you to look at is based upon the Healthy Places Index, you get a number. Then they break it into quartiles. So the bottom quartile represent the unhealthiest areas, and the top quartile represent the healthiest areas. This is a map of San Diego. We actually use this to implement testing strategy within San Diego. The heaviest populated area and the most underserved populations are in the southern part of San Diego, going down by the border. You can see these black dots. These are all test sites. One of the things I was involved in is making sure that our county opened up enough test sites in the underserved areas before they started opening up in this area up here. And the county was very responsive and had some success. So these dots represent test sites. What we're hoping is that when we start implementing the vaccine strategy to the underserved population. They would use the same strategy so that people of Latinx, Hispanic, Asian, Black in these low-income areas will be priority as far as getting the vaccine. So good question is, what do Black folks think about COVID-19? Several months ago, a group did a survey of African Americans, asking them questions about what they thought about the vaccine. It was sponsored by the Equity Research and Innovative Center at Yale School of Medicine. I think that Dr. Marcella Nesbitt, I forget her last name, Smith, who's on the Biden transition team, was part of this. But basically, when you think about coronavirus, now these are all Black folks, what one or two words come to mind? And you can see scary, death, dangerous, sick, contagious. So this is just letting you know, Black folks have not bought off into this is not something to be concerned about. They realize it's significant. Now, how are you responding? Are you taking safety precautions? How often do you follow these recommendations regarding protecting yourself and others from COVID-19? Frequently washing hands, staying at least 6 feet away, avoid touching your eyes, nose, and mouth, wearing a mask. Now, as you can see, Black folks are doing pretty good. So they're not responsive. It's very difficult for everybody to avoid touching your eyes, nose. I know I am. So I think Black folks in general are very in tune to what's going in and trying to do the right thing. This is about wearing a mask. Overall, this is by age, 18 to 34, 35 to 49, and then 50 to 64, et cetera. You can see as a community, Black folks are very often to feeling safe about wearing a mask. I find this very interesting, that no, I do not feel safe wearing a mask. About a quarter of the 18 to 49. Now, I think this has something to do with Black Lives Matter. I mean, can you imagine a Black man, young Black folks now wearing a mask, walking down the street or near a bank or something? So they're very concerned about what's going to happen with them in our society. At least that's my interpretation. The other is we hear a lot of argument here in San Diego, and I guess elsewhere, every time the governor or somebody comes up and says we need to shut down. And there's a lot of protests many times about shutting down businesses. Why do we have to shut down the business? It's more important that the business is open. Some people say it's time to end and shut down, reopen, etc. 80% of Black folks realize that there's an issue with shutting down businesses, losing jobs, etc. But I was kind of surprised that it was this high. They're are very supportive of holding off shutdowns to assure safety. And I think this is my last slide on this. The other thing is, for each of the following statements, indicate whether you agree or disagree. Black people are less likely than whites to be offered coronavirus testing. Black people are less likely than whites to get admitted to the hospital for coronavirus. Black people are less likely than whites to be offered experimental treatment for coronavirus. Black people are less likely than whites to have everything done for them. 60% or more of Black folks feel that there's a lot of discrimination going on with Black folks when this comes along. That shouldn't surprise us. It goes along with our whole distrust of the system. And I think data bears that out. That is true. So, who do we trust? Here's our dilemma. Black, Latino population have the highest rates of COVID, deaths, hospitalizations, yet we have the highest rates of vaccine hesitancy. This is from the CDC, really just showing when we're talking about hospitalizations, we're talking about deaths, Blacks and Latinx are at the top. When we talk about comorbidities do not cause you to get coronavirus, but if you get it, you're at a greater risk of winding up in the hospital and death. And this is just showing the risk of hypertension versus having 2 or 3 more comorbidities. And guess who is very high on the scale of comorbidities? We are. So we're at greater risk because of this, and probably need to be the first in line to get protection against this virus. But at the same time, want to wait in line to see what happens to everybody else. So this is also another survey asking Black adults have less positive views of the medical scientists than Hispanics and whites. The question is the percent of US adults who have a great deal of confidence in the medical scientists to act in the best interest of the public. 35% Blacks say they do, where about 10% more are white and Hispanics are the same. Have a mostly positive view of medical research scientists. About half of Blacks are there, 2/3 of Hispanics and whites. Have a mostly positive view of medical doctors, 60% for Blacks and higher for Hispanics and whites. So you can see we're the most reluctant. We have issues, historically justified, but we have issues. This is critically important because in May, this is looking at gender, age, race, ethnicity, and education. And those who said they would get a COVID-19 vaccine was higher in May and significantly went down by September. What was happening? I think the mixed messages coming from the White House, coming from the CDC, coming from everywhere, really led the population in general to be less confident in any vaccine coming out, but especially Black folks. We were maybe halfway there, now only about 1/3. So whenever something happens, it affects us much more severely. This was really the purpose for the National Medical Association forming this NMA task force, realizing that the confidence level in the Black community was going down. We wanted to have independent Black scientists evaluating the data to see if we could prove that. So this brings me to Black folks' dilemma. Now, many of you know me. I give a talk on racism in medicine, and everybody talks about the Tuskegee syphilis study, but you and I know there are multiple studies that have caused us to get to this point, besides syphilis, going back to Marion Sims, who operated on slaves and Black women, and multiple inhumane things that were done to Black folks. But a lot of folks, all they speak about is the Tuskegee study. I find it ironic that we have a drug company that discovered a drug called penicillin that was a cure for syphilis, but the cure was purposely withheld from hundreds of Black men infected with the syphilis. Today, we have a drug company that produces a vaccine that makes you immune from the deadly COVID-19 disease, but the historic distrust resulted in the victims now themselves withholding the protection. I don't know if that's white man's reverse psychology, but now he doesn't have to withhold it, we are. I can tell you my colleagues, especially my white colleagues, are lining up trying to get this. And I think some of us should be in line first, but because of this resistance, we're giving up our spot. Now, real quick, I saw a cartoon sent to me about the Pfizer vaccine. Then I had a conversation with one of my patients who had called on the phone, and he wanted a refill of his medication. He wanted his medication with that blue pill. He called a Viagra. He said, "Don't do it. I need a refill of my Viagra." And I said, "Okay, no problem. I'm going to take care of your blood pressure pill, give you a few of these." And I said, "Now, what about that vaccine?" I was trying to get him to take the flu vaccine. He said, "Nah, doc, I haven't had that flu vaccine. Wow. I don't know about that other vaccine either. I ain't taking that." I said, "So you're talking about the COVID vaccine? You don't know if you're going to take it?" He said, "No." I said, "Well," I said, "do you know who makes Viagra?" He said, "No." I said, "Pfizer." I said, "Do you know who makes the new vaccine that just came out?" He said, "No." I said, "Pfizer." I said, "So if you trust Pfizer with your erection, why not trust Pfizer with COVID?" Stay safe, protect, get vaccinated. Maybe only Dr. Hood could get away with this, but I use that with my patients. I've never had any of my Black patients ask me, what's in that Viagra pill? What's it mixed with? Is it, what are the long-term effects? All they wanted to know was, could they get a prescription and where could they get it cheaper? Yet everybody wants to know what's in this. So I heard you, Dr. Maxey, talking about something similar. You've got to be practical. This is something about trusted messengers for Black Africans. This was part of that survey. They asked, who do you trust most about factual information on COVID? At the top were doctors and nurses. I've actually seen another survey where Black doctors and Black nurses were even higher. But local public health officials, Black elected officials, Dr. Fauci, civil rights organizations, Democrats in Congress, Dr. Jerome Adams, this is a public health announcement, but some of my lost colleagues, they rely on Fox, Republicans, and Donald Trump. So with that, what is the vaccination strategy for the African American community? My answer is, in the 1930s, we didn't have these great Black scientists. That was the past. Today, we got Dr. Hildreth, who is President CEO, Meharry Medical College, Professor of Immunology. He's on the Biden-Harris COVID Advisory Committee and also a member of the FDA committee that voted on the Pfizer Moderna vaccines. We got Dr. Abugu, who is at the Yale School of Medicine, professor who works with viruses. We got Dr. Adam Mora at the University of North Carolina, who is professor of epidemiology, infectious disease, HIV. She says you should take it. On the NMA task force, we got Dr. Sonia Hutchins, who is at Morehouse School of Medicine, professor of community health and preventive medicine, also a member of the NMA task force in California. Almost through. I'm sorry. In California, we got our own Dr. Oliver Brooks, who's at the Watts Healthcare Center. He's also appointed by the governor to one of the task force here in California, Dr. Virginia Kane, infectious disease. She heads the National Medical Association infectious disease section of the NMA from Indianapolis, head of the Department of Health there. She's part of the NMA task force. We got Dr. Dio Hewitt, who is an infectious disease expert in New York, spent about 15 years doing clinical research in the pharmaceutical industry. We got Dr. Patricia Whitney Williams, who actually represents the National Medical Association on the ACIP committee. And she is a chief of the Department of Pediatrics at Robert Wood Johnson Medical School, infectious disease. And who I love is Kizzy Corbett, who's a viral researcher at the NIH under Dr. Fauci and Dr. Leon McDougall. So we didn't have this during 50, 60 years ago. We got a lot of Black folks who look like you, think like you. We're very concerned about the same things we are. And so my answer is, what's different then than today? We are. Lastly, how we traditionally communicate with hugs, handshakes, and dap. All of this is being changed. I think we need the acts of connectiveness, intimacy, expressions of affection and admiration, unity, but with this social distancing and this virus, it makes it difficult. How do we get back to that? A strategy should be for the Black community: love, knowledge, and understanding. We fall into three groups. There are those Blacks who say, I'm ready. Where can I get it? The only issue here is making sure they get access to it, and it doesn't cost them anything. We got the vaccine hesitancy group that can be reached by targeted outreach, education, using scientific data, using trusted messengers, engage faith communities, sororities, fraternities, and of course, healthcare workers. And yes, we have vaccine objector almost no matter what we say for holistic reasons, religious reasons. They may say, no, we need to love them too. We need to encourage safe mitigation, healthy lifestyles, physical distancing, limit travel, avoid crowded spaces, wearing masks, sleep, exercise. I believe in vitamin D, C, and Z, and eating healthy. In San Diego, we have taken this to heart. There's an advertising campaign specifically at the Black community. Now we've got over 50 billboards throughout the city, not only talking about AIDS, but tying into social change, social justice. Social change starts with surviving COVID. This is a TV ad that comes on every day.
Amazing grace, how sweet the sound that saved a wretch like me.
So that I can share. Okay, that's it. So thank you. Black COVID Facts SD is the website that we have specifically for African Americans with messages, videos from trusted messengers trying to get Black folks to do the right thing and take the vaccine. Thank you.
We thank you so much, Dr. Hood. We appreciate your stellar, as usual, presentation. I know we have questions for you, but I want to ask the first question. Earlier this week, myself being a frontline physician still practicing, being a senior in the above 75 years of age, I had questions. What is the availability of the vaccine to someone such as myself and my staff? But in general, what is how can someone find out where they are on the list, if they're not in a nursing home, etc.? And I understand most things are controlled through the counties. How would you advise people to find out where they and their relatives stand?
Yeah, so that's an excellent question, and you're right. Most of it is held through the county, so you really need to kind of check with the county because most counties are trying to follow the same thing that came down. However, for specifically to you, physicians need to register. There is a immunization registry, or this is actually a requirement of the state. So you need to be registered with them because they want to know who's getting it. And then once when you registered, I'm told it takes 3-4 days to kind of get approved. Then you get put on the list. And so as your tier comes up, they supposedly will let you know where you can go to get that vaccination. I can tell you here in San Diego, they just came up with a website that is helping physicians with that you can go to that website and see I'm in 1A tier 2, and they will say in the next 2 weeks, you will be eligible and this is where you can go. So I'm not sure what they're doing in Los Angeles. It's not centralized like I think it should. And that's not just for you, but when you register, you should be registering you as well as your staff.
Well, thank you. And I'm going to ask some of our steering committee and advisory members to help us find this information since we have people here from all of our states and put some of this information on our blackhealthtrust.org. Dr. Neighbor, are there any questions for Dr. Hood in the chat box?
Yes, there are several questions Several of them, though, I'm going to hold until we finish because I'm pretty confident that they're going to be addressed in one or the other of the following talks. But there's one specifically to Dr. Hood, and I tried to paraphrase it, but I think it'll be clearer if I just read it to you, Dr. Hood. The ACIP model contains equity.
I think you're muted.
She just froze.
Into assignment, can you read that?
Sorry.
Sure.
Into or Simon, can you read the statement he was reading?
Sure.
Well, I can see it. I can read it. Is it the ACPI model contains equity issues which LA County is wrestling with this right now? 1A allows, it's a very long question. Let me put it to you like this. I think I presented a model called the Health Equity Matrix using the Healthy Places Index. I think if you look at it as a model, it looks good, it sounds good, but implementation of it is really in the eyes of the beholder. And I think that places are going to have problems struggling with how you implement that if there isn't some type of consensus. I know that's what the local vaccine advisory group is doing in San Diego. I'm one of the tri-chairs, and one of the things we're struggling with is just this issue. When we get to the point, especially when we start going to the minority populations, how do we make sure that the folks who want to get the vaccine in those areas get it? So we're looking at what vaccine is available, where, who's going to be doing the vaccination, what are the places in those areas that will be doing the vaccinations. So this is what we're doing in San Diego.
Okay, well, thank you, Dr. Hood. Hopefully you'll stay around. We're going to have a lot of more questions, and we're going to speak to all the speakers. Before we go to our next speaker, I want to again say to everyone that we are providing you with information, but we specifically are not giving attendees individual medical or healthcare advice. We want you to take your questions and get answers from your local provider and make sure that these answers are specific to you. For those people who may not have a primary care physician or a provider locally, we ask you to go to biometrics-second-opinion-experts.com, and we have physicians who will provide you good advice that is medically sound, and it will be as a service at this time. But here on Black Health Trust, we're not giving legal medical advice, even though you ought to listen very carefully to our selected speakers. I'd like to introduce for a brief statement the chairman of our advisory committee, Dr. Carl Brooks. Mr. Brooks.
Good afternoon, everybody, and Happy New Year's to everybody. The impact of COVID-19 on the world has been dramatic. I'm 70 years of age, and I talk to my 96-year-old mother every day, and she's not experienced anything like this in a lifetime. So we're really going through something that is major and is impacted and is real. The poor are being affected. We're seeing people searching for food and getting handouts and things like that that have never been in this situation before. And I live in Arlington, Virginia, and it's considered a high-end kind of community. There are lines all around my community with people taking advantage of this because they need the goods and services. My friend and brother, Dr. Randall Maxey, had a vision, and Randall and I have been tight for a long time and will be tight for the next 100 years. And his view was that we needed to have a African-American response and understanding of what was going on, and we needed to be able to get it to our people. The impact of these programs has been tremendous. You know, we range somewhere between 7,000 and 10,000 people listening to our program every Sunday. We're getting an enhanced website that'll be available around between the 8th and the 10th. That's going to really be first class. It's going to allow us to really package and put the information together in a super way. These Sunday programs have gone on for every Sunday, despite football and despite all the things we like to do, have gone on since May, June of 2020, and will continue. These are outstanding. We got the access to outstanding doctors of color and doctors and supportive doctors not of color who have supported us and provided excellent information. As Randall says, we're not advising on you medically. We want you to check with your own doctor, but they've done a wonderful job of providing that information to us. I'm asking a couple things. I'm asking all of you that live in the Georgia area to go out and vote. The politics in our country are crazy, absolutely crazy. And we need the help from everybody that has the ability to vote in Georgia properly to vote and get your vote out there so we can be able to move the Congress to do the right things as we move forward. Frankly, you know, if we're to continue to do this program as we've done it since May, June to now, we're going to need financial help. The truth of it is that I'm a former corporate executive at Boeing and General Public Utilities, and I ran the Executive Leadership Council. So I know all about making money, and I know about working with excellent people. And the group of doctors that have been assembled to speak to us is world class. I used to call the ELC members the baddest brothers and sisters on the planet. This is a bad group of brothers and sisters that are helping our community in a positive way. So we need your help. We need your help in order to donate so that we can do this in a world-class way. We need more administrative support. We need backups. We need additional computers. We need all the things that companies need to take this to a world-class platform. And I'm sure that that'll happen. We recognize, and there are no set amounts, and you can contribute any amount to be helpful to us. Before I close, I want to make you aware of a program that's going to be coming on One Shot, and it's going to be coming on both Facebook and YouTube on Thursday, excuse me, let me be real clear. On Thursday, 7 to 8:30 Eastern Standard Time. And it's going to be a town hall meeting on the vaccine, the virus, and they're going to be familiar people to all of us that have seen the doctors who presented to us. Dr. Fauci is going to be on this program. We're going to have Dr. McDougall that was on with us last week from the NMA, and they're going to discuss what's going on, the vaccines, and the world is recognizing that we have a reluctance. And Dr. Hood explained it in all kinds of ways. We have reason to be paranoid, and so this program, hopefully with what we're doing every Sunday, will help us to gain the confidence to do what we need to do. We can either get the corona or we can get the vaccine. I mean, it's that simple. And so, as was said, I'm going to log in and make sure I get access to it for me and my family. I got a 96-year-old mother. I got a wife that I've been married to forever. And we got to make sure that we're around the party with our kids and grandkids. So we got to do the right thing also. With that, I want to sign off and wish everyone a healthy and safe 2021. We're going to have a big party when this thing is over. It's going to probably be nationwide, but we're going to have a big party when this thing is over. I appreciate the support. We need your financial support, and thank you. And we love you all.
Thank you so much, Carl Brooks. And I have to welcome back Dr. Barbara Neighbor-Stevens, apparently in Tennessee. They had a power outage for a few minutes. Welcome back, Dr. Neighbor-Stevens. I want to do something abnormal for just a moment, and I hope you will cooperate with me. As Mr. Brooks was talking about the fantastic physicians, I would be remiss if I didn't recognize the stellar physicians that are on this call. And Simon, I want you to unmute this for like 3 minutes, and I want the physicians who are on just to say their name and their specialty, no more, just to get a flavor of who we have in addition to our regular listening audience. And I'll start out, just introduce yourself, Dr. Sherrod, name and specialty.
Jesse Sherrod, pediatric infectious disease, a former ACIP member.
Next, I see Dr. Richard Williams. We got to do it quick.
He's gone.
Dr. Ivan Walks,
Dr. Ivan Walks, public health and psychiatry.
Dr. Helen Davis,
Helen Davis, ophthalmologist, member of American Academy of Ophthalmology.
Dr. Walter Fajay, Dr. Michelle Clark,
Michelle Clark, psychiatrist, member of Black Psychiatrists of America, National Medical Association, and Black Health Trust.
Dr. Alice Coombs,
Dr. Coombs, chair of the section of anesthesiology for the National Medical Association. Thank you.
Dr. Richard Williams,
Dr. Richard Allen Williams, past president of the National Medical Association and cardiologist on the UCLA faculty.
okay, help me out, Dr. Neighbor Stevens, I can't see everybody.
Dr. Villanueva,
Hi, Rachel Villanueva, OBGYN in New York City, affiliated with New York University, president-elect of the NMA.
Yay,
Dr. Rogers Kane,
Dr. Rogers Kane, family practice, Jacksonville, Florida.
you got anybody else?
I don't recognize anybody else. If I missed anyone, please jump in quickly.
I think Dr. Vicky Pace is on from UCLA Psychiatry.
And Dr. Monique Hedman-Maxey, family medicine intern and future geriatrician in Los Angeles.
Are you
I'm
related?
Dr. Wendy Green in Los Angeles, a past president for the Charles Drew Medical Society and for the Association of Black Women Physicians. And
yeah, and Dr. Walt Baguette, assistant professor of family medicine, Howard University, Washington, D.C.
Dr. Michael Carter. Dr. Michael Carter, just on the phone. Functional medicine doctor, chief medical officer for Health Revival Partners.
I see Dr. Motley.
Yes, Dr. Motley. I'm a medical MD, PhD, and a developer of medical devices and treatments, and I'm in Los Angeles.
Okay.
Dr. Carolyn Towler, diagnostic radiology and integrative holistic medicine, also in Los Angeles.
Dr. Lisa Curvin Dawes,
internal medicine, infection control, working with the COVID team for Jamaica.
I thought I heard a Southern accent. Any other physicians on the call? Well, thank you very much, my colleagues. I'd like to ask Dr. Neighbor to introduce our next speaker, Dr. Corin.
So Dr. Corin is our cardiologist in Jacksonville, Florida, working on clinical trials, who will discuss with us further issues around the vaccines and will address the issue of emergency use authorization versus traditional path to approval. Dr. Corin.
Well, thank you very much for inviting me. Thank you for actually enlightening me. I love this conversation. Happy New Year to all. And I look forward to having a great discussion after my presentation. I hope everybody can see my screen okay, but I will direct my slides and try to move through this relatively quickly to give us more time for questions and answers. I actually had some thoughts about Dr. Hood's really tremendous presentation and like to maybe share some concepts that are a little bit off topic for me in terms of my prepared comments, but I think would be enlightening for folks. So the approach that I took for my talk was to start with the basics and then move us quickly to why it is that we are fortunate to have these new technology vaccines available and why all of us, particularly the Black community, should access these incredible new levels of technology. So to start, in the most basic terms, a virus is simply a piece of bad news wrapped up in a protein. I really love that description. And a virus is the most basic life form. It's so simple that it can't actually live standing alone. It requires a host. So it has to infect another living cell in order for it to propagate itself. And so when you think about that, you get some insight into the fact that viruses have a way that they have to go about their business to propagate. And if you understand the way they go about their business, you can also understand the ways to prevent them from causing disease. So there are many types of viruses that exist, and they have very different characteristics. And you can see here, the cartoon shows a typical coronavirus with its characteristic spikes.
Doctor, you need to share your screen.
Oh, you can't? I'm sorry. Okay.
Can you help, Simon?
Yeah, tell me if folks are able to see it.
Not yet.
Yeah, alrighty. Let's see if this works. Can you see it now?
No.
Hitting share screen. Multiple participants can share simultaneously. Let's see. Tell me if this works. Can you see it now?
Yes. Yes.
Okay. Okay. So you folks missed the first slide, which is basically actually the first slide is just a picture of me, which can go by very quickly. Second slide is just a picture of a coronavirus, and this quote from Jean and Peter Medawar, and then getting here. So there are many types of viruses that exist, and they have different characteristics. And understanding these characteristics is quite important in terms of fighting the viruses and preventing disease. And just for some of our lay members, I'm just throwing out some of the names. And one of the reasons it's hard for people to understand some of the nuances of fighting viruses is because just the names, the nomenclature can be quite confusing. So you've heard of hepatitis viruses, but hepatitis viruses are actually different families of virus that all have in common the fact that they affect the liver. And hepatitis B and C, for example, have completely different ways of being approached and treated and attacked. Other viruses that you heard of are herpes viruses, smallpox viruses, influenza viruses, and of course, most acutely, we are hearing a lot about coronaviruses. And coronaviruses are a family of viruses that are characterized by these spikes that come out from the sphere of the virus. And these spikes are quite important in terms of their pathogenesis or their ability to infect human cells, which I'll talk about in a moment. So also to just help people clarify their understanding of viruses, when you use the term influenza or use the term coronavirus, you're actually talking about the family names. These are family viruses. But when you get to specific viruses, you're saying things like the H1N1 flu or COVID-19 or SARS-CoV-2. Now you're getting into very specific elements of a particular virus. And that's important because there are some characteristics of viruses that are common to families, but then there are characteristics that are specific to specific viruses. So for example, what's characteristic of both influenza and coronaviruses that's true to all the members of the family is that these are viruses that contain RNA. The instructions for how these viruses go about their business is based on RNA. However, for coronaviruses, the vast majority of coronaviruses don't infect human cells. They would typically infect bats or pigs or other species, but they rarely infect human cells, in fact. In fact, only 7 coronaviruses have been shown to infect humans. And of the 7, 4 really cause very mild symptoms like the common cold. But 3 of them, which are SARS, MERS, and now COVID-19, have much more significant pathogenesis and cause much greater problems for humans. So to remind everybody about these things, again, just to start from the basics and then move to the more complex, is that Chinese scientists first discovered COVID-19 related to an outbreak in Wuhan province in late 2019. The virus is believed to probably have started in bats, and it mutated in a way so that it jumped to humans probably sometime in late 2019. And at this point, the virus has spread to all parts of the world, largely through travel. We are a mobile society, and people have traveled. And in fact, one of the reasons that the virus spread about the way it did was based on travel to and from China initially. So Italy, which was an early epicenter, has a lot of commercial ties to China, and that was part of the initial wave. And certainly China and the US have multiple ties, and many people moving back and forth. In the early days, when there were restrictions placed on people from certain countries, what didn't happen is that US citizens who were visiting abroad were allowed to freely come in. Unfortunately, these people ended up spreading the virus throughout the United States. Obviously, there are trade-offs with these type of decisions because we certainly don't want to prevent people from coming home. On the other hand, they were in endemic areas and did bring the virus to the United States. I think we all understand that COVID-19 has led to an absolutely unprecedented interruption of the world economy, and also an unprecedented rapid medical response, which has been just absolutely dynamic and absolutely tremendous. And I've had the good fortune to be part of it. Just to let people know some of the detailed elements of my involvement, I was the principal investigator for both the Pfizer and the Moderna vaccine studies here in Northeast Florida. And I'm currently an investigator in the Novavax study, which is actually enrolling patients as we speak. And I'll speak about all these things in more detail momentarily. I think we all know that COVID-19 has been absolutely devastating. Over 80 million people around the world were diagnosed with COVID-19 in 2019, and more than 1.8 million people died. In the US, there have been now 360,000 people who have died of complications of COVID-19. And you can see a graph on the bottom of this slide that shows the countries that were hit hardest by the virus in some of the recent statistics. So moving, sort of fast forward moving, we realized early on that probably the most important way to combat this virus is to develop a vaccine. And historically, vaccines have taken many years to develop. In fact, a typical timeline for a vaccine development would be 3 to 5 years. But fortunately, with this emerging technology that we have access to, we've been able to compress that in ways we didn't think were imaginable. We, in fact, compressed that timeline to months. And I would even predict that based on what we've learned from COVID-19, if there's another similar outbreak of another virus, we may be able to compress vaccine development to weeks, which is tremendous when you think about it. Over 150 COVID-19 vaccines are in development and have been registered to the WHO. And about 20 of these have been manufactured and tested in humans already. Of the vaccines that have been manufactured and tested, there are 2 that have received emergency authorization in the US. And I know there are some questions about that, and I'll address that now. The 2 that have received emergency authorization are Pfizer BioNTech. Pfizer partnered with a German biotech company called BioNTech, and Moderna, which is a US-based company, have created vaccines that use something called the messenger RNA platform. And both of these have received emergency authorization in the US. Emergency authorization is different than approval. So when a manufacturer gets a drug approved by the FDA, a manufacturer is able to sell their product to pharmacies and to physician offices and to hospitals, et cetera. With emergency authorization, the manufacturers can only distribute the drug through the government. And the reason that was done is because the FDA took the unprecedented move of approving these vaccines based on 2 months of data. Now, they did require the companies to look beyond that. And in fact, we have some data beyond 2 months. But because of the urgency of the situation with COVID-19, the FDA did approve emergency authorization for both the Pfizer BioNTech product and the Moderna product after 2 full months of data were obtained. And this effort was just truly unprecedented. So as mentioned, the Pfizer BioNTech study involved 44,000 people who enrolled in less than 4 months, which was just absolutely tremendous. And the Moderna program ended up enrolling about 32,000 people. These are massive, massive clinical trials. And the amount of information that we've gotten from these clinical trials is just absolutely off the charts. But on the other hand, this is only emergency authorization use. So that means that the average person cannot go down to their local pharmacy and get these products. You can only get it through some government-sanctioned distribution system. There are other products that have been actually approved in other parts of the world. So it was mentioned that the Oxford AstraZeneca product, which is a modified adenovirus, where the adenovirus is a vector for expressing the spike protein, has been approved in Great Britain and some other places around the world. This has a published efficacy rate level that is less than that of the Pfizer, BioNTech, or the Moderna products, but still reasonably effective, between 70 and 90% versus 95% for the messenger RNA platform viruses that I'll show you in a second. The advantage, though, of the Oxford AstraZeneca product is that it can be stored at room temperature, whereas the others require freezing or deep freezing and refrigeration for a period of time before they're reconstituted and ultimately given to patients. So a potential advantage of the Oxford AstraZeneca product, even though it's probably going to be less effective, is that the distribution logistics should be simpler. Another one that you've probably heard about is the Sputnik vaccine that also uses an adenovirus vector, which was developed by the Russians. The amount of clinical trial testing that was done before Russia approved this was a very, very, very small fraction of what we expect in the US and in Western Europe. In fact, there are just several hundred patients that were looked at before it was approved. The Chinese have also developed a vaccine that has been approved in other parts of the world that involves injecting a virus. But again, these products may be good. We just don't have nearly as much data, but they will probably be distributed to parts of the world that don't have the resources to deal with the logistics of the messenger RNA platform vaccines that I'll be talking more about in a moment. So I think it's probably worthwhile to just get a sense for how different the messenger RNA platform is from traditional viruses. So the next couple of slides, I'll briefly go through how traditional viruses are made. So for traditional virus, for traditional vaccine, first you have to grow the virus. So you would have to grow a small amount of a specific virus in some sort of cell culture. As I mentioned, viruses can't exist outside of cells, so you have to create an environment so that the viruses can reproduce themselves and ultimately reproduce themselves in great numbers. There are different cell types that can be used. Typically, we would use a chicken embryo, eggs, or cell lines such as tobacco leaves that can be leveraged in a way that the viruses can grow very, very rapidly. Unfortunately, when you have that particular way of developing vaccines, you also have the problem of separating what you want to inject into people from the media that's used to actually grow the virus. So this next slide shows that for step 2, you have to figure out what antigen or what protein that you'll use from the viral cells, and you have to separate that from the material used to actually grow the viruses. And the goal here is to release as much virus as possible, but it's a very challenging process in order to get the actual protein or the part of the virus that you want to inject in people and separate that from the rest of this huge vat of a virus culture that's being manufactured. Then there are further efforts to purify the antigen. Obviously, you want to inject into people things that are as pure as possible. So there are newer technologies that have helped us move forward on this particular step, including using recombinant technology processes that will produce just the pure antigen once you know what that antigen is. Step 4 involves making sure that whatever you give to a patient is completely inactivated. The last thing we want to do is to give somebody a virus that can reproduce itself. And as I mentioned, the Oxford AstraZeneca product uses an inactive adenovirus vector, which is you're actually injecting a virus, but you're injecting a virus that can't reproduce itself, that is ultimately just a vehicle to reproduce the spike protein. And other types of vaccines have tried to leverage these type of approaches. And then finally, you have to add some sort of adjuvant or material that enhances the immune response. And that way, the immune system can recognize the antigen that you're using as quickly and as easily as possible. These vaccines also usually have stabilizers and other things to prolong the shelf life, and preservatives to allow them to be kept at room temperature and to be distributed around the world. And obviously, with any product, there is a major manufacturing process. You have to get the stuff into vials, you have to get the stuff into trucks, and then get the vials that are in the trucks to your local pharmacies, et cetera, so they can be given to patients. So a big part of any distribution program are the logistics of actually getting it to patients. As I mentioned, with the messenger RNA products, this logistical chain is a little bit more complicated because it involves doing this all in refrigeration or freezing. But even with those logistical challenges, the science of messenger RNA is really just spectacular. I couldn't be more excited about it. So here, the concept is rather than injecting a virus's antigen or protein into your body, the messenger RNA vaccine gives your body the genetic code needed to produce that antigen itself. It's teaching the body what to do. So In fact, what happens with messenger RNA is when the antigens appear on the outside of your cells based on this set of instructions, your immune system recognizes these proteins as foreign. Your immune system is very smart in that way. And because it recognizes them as foreign, it attacks them, and then it learns how to defeat future intruders in the process without ever giving the virus or without ever giving a piece of the virus. It's purely a set of instructions. And I made my own sort of editorial comment about that, which is, I think this is just the absolute coolest technology, is that you turn your body into its own vaccine manufacturing unit. So one of the things that has come up quite a bit is this concern about messenger RNA. And RNA sounds like this high-tech concept, and it is in a lot of ways. But what people don't necessarily understand, it's actually the way your body naturally functions. Messenger RNA is a messenger. So the messenger RNA is the way your body takes information from your genes and conveys it to other parts of your body so your body knows what to do. And at any given time, there are literally thousands of different messenger RNAs that are communicating information to your body. So it's actually the way your body naturally functions. And the other thing I find is useful is that messenger RNA doesn't change your genes. I've heard a lot of people concerned that it will change our genetics somehow. So you can see, and these are some terms from high school or college biology, but on the left side of the slide, you can see that we're looking at DNA, and that represents your genes. That's in the nucleus of cells. And then on the right side of the slide, we have the proteins. And these are the action molecules. These are what makes up hormones and other things that gets our body to do what it needs to do. And between them is the messenger. So the messenger, here now circled, doesn't actually even go to the part of the cell where your genes are contained. This is in the cytoplasm, and this helps our body just create these proteins, which then primes our immune system to fight COVID-19 or whatever the virus may be. So it is super cool technology that actually doesn't get into the DNA, but it is RNA. The other thing that's interesting is our bodies have natural defenses against viruses, and those natural defenses against viruses use RNA. They use something called small silencing RNA to recognize sequences that are foreign sequences and then block those sequences. So again, it's kind of funny when some people don't want to use a vaccine because they perceive it to be not natural, but these vaccines are actually exploiting completely natural body functions. The other thing that's important in terms of understanding is this concept of antibodies, both non-neutralizing and neutralizing antibodies. So one of the advantages of the new technology is you can be very, very specific about the antibodies that you create. So on the left side of the slide is kind of what happens if you just get a virus. If someone gets a virus because they get sick, your body is creating all kinds of antibodies. Some of them will neutralize the virus, and some of them will just stick to the virus. So just to walk you through this cartoon a little bit, that kind of bluish purple blob in the middle is the cell. And you can see that the cell has a Y-shaped thing that comes out of it. That's the receptor. And the little green house is the virus. And the point of the house is the part of the virus that gets to the receptor of a human cell, which will ultimately allow that virus to get into the cell and reproduce itself. And those little grayish triangular molecules are the antibodies. So if the antibodies are sticking to the virus, but don't block the virus's ability to infect the cell, they're not doing a whole lot. But with the messenger RNA platform, we can create neutralizing antibodies that stick to the point or the place on the virus that actually is responsible for getting into the human cells. This is important from a number of standpoints, including this concept of mutations of viruses. Because if the point mutates, the likelihood is it will not be able to infect human cells. And if another part of the virus mutates, it doesn't affect the ability of the messenger RNA product to block infection. So this point was made earlier, but I think it's really important. There's only two ways to become immune to viruses: either get the disease, recover, and mount a response that includes antibodies and cell-mediated immune activation. So, for example, if somebody gets the measles, you get sick, hopefully you don't get too sick, but you're immune for the rest of your life, but you had to deal with the complications of the disease, including, for many viruses, long-term complications. Or you can get a vaccine that creates antibodies that kill the virus and elicits a cell-mediated response that protects you from the virus in the future, by training your immune system what to do. And a good example of this is getting the COVID-19 vaccine, which protects us against COVID-19. Well, how do I know this? Because we have amazing data at this point to show us exactly what this vaccine does. So this comes from the New England Journal of Medicine. It was published last month, and it really summarizes everything that I've just said. On the left side of the slide, right under stunning data, you can see the messenger RNA platform in a small lipid nanoparticle that helps the messenger RNA get to the part of the cell that it needs to get to. And then it teaches the cell, again, not in the nucleus, but in the cytoplasm of the cell, to produce the protein that induces the immune response, which you can see right under that little cartoon that says spike protein. And then if you look to the right side of the slide, you see what I consider absolutely stunning. So if you look at people in the studies that I participated in the blue are people in the placebo arm, in the red are people in the vaccinated arm. And you can see that people in both arms were equally likely to get COVID-19 for the first 7 days or so after vaccination. But starting at around day 10, these curves completely diverge. And people, this is after the first dose of the vaccine, have close to a 0 likelihood of getting COVID-19 for the time period of the study that was evaluated. Whereas people in the placebo arm continue to slowly but steadily get COVID-19. It was about 2% of all patients that got COVID-19 after this 2-month period of observation. And if you look at severe cases, people who are vaccinated, by the way, this looks at Pfizer data, but it's also true for the Moderna data. The likelihood of getting a severe case of COVID is really close to 0 for people who have been treated with these vaccines. So these are just absolutely stunning data. And I will tell you that the first day that I could get the vaccine, I got the vaccine. I received the Pfizer vaccine the first day available. I would have gotten the Moderna vaccine because I'm equally confident in that. And it's really just breakthrough science that we should be highly supportive of, in my opinion. So I'll stop at that point. Again, I'm really looking forward to questions and stimulating conversation from here. But again, thank you for inviting me, and thank you for the opportunity to let people know why this is such an important breakthrough in medical science.
Thank you so much, Dr. Corn, for your outstanding talk. While Dr. Nabert looks at the chat box, I'm going to ask you to review something. There are a couple of words I want you to define in your explanation. One is vector, and one is the messenger RNA. And
Sure.
is it clear, and I know you said it before, that is the analogy correct? This is almost like 3D printing, that you're not giving the actual virus. Can you just walk us through that very briefly?
Sure. Yeah, so with the messenger RNA technology, you are not giving the virus or anything from the virus other than a code to tell your body how to make a protein that is equal to the part of the virus that infects human cells. So you're telling our body, this is the bad sequence of proteins, which could be, they're actually particles, they're nucleotides is the proper word for it. These are a series of nucleotides that will enable this bad guy, this bad virus, to get into our cells. And because you have seen this before, you are prepared to fight it. And that's how our bodies work. Our immune system will then create what's both antibodies and what's called cell-mediated immunity to identify the bad guy, this sequence, and then send out antibodies to block that sequence from infecting human cells. And then to get to your first question about a vector is a strategy that other vaccines have used, such as the AstraZeneca Oxford. And basically, you create something that is used to carry the information to a place where it needs to go. So we use the word vector sometimes. For example, a mosquito is a vector for disease. So it doesn't cause the disease directly, but it will have a pathogen that it gets into its body and then injects into a human. So the mosquito is the vector. And in the case of these vaccines, the adenovirus is the vector. It's the way that we get the information to our bodies to ultimately produce a spike protein or produce whatever it is that we're using to allow the immune system to recognize the bad guy. So a vector would be characterized as one of these adenoviruses or other ways of getting that information to the body. Hopefully that's clear.
And it is. And my last question before Dr. Stevens comes up with one other question from the chat box. The relative risk of side effects versus COVID from these vaccines. Would you just briefly review that again?
Sure. So fortunately, I didn't go into the side effect data, but it's very, very favorable. So after the first dose of the vaccine, the side effects are really quite minimal. And there's a couple of interesting little tidbits here. One is that younger people tend to have a little bit more of a side effect profile than older people because the side effects related to activation of the immune system. And in younger people, your immune system may be activated a little bit more vigorously. But the type of side effects you get would be low-grade fever. You would get some maybe chills, some muscle aches, that sort of thing. When I got the vaccine, the only thing I experienced was some soreness in my deltoid for 12 hours, and that's it. Now, for the second dose, because your immune system is already activated to some degree, you may have a little bit more of these symptoms. But again, the likelihood of severe side effects is really quite minimal. On the other hand, from COVID, you have, particularly if you're older, your likelihood of being hospitalized is in the teens. Your likelihood of dying is not insignificant. I can go over those data in a second. And so you're looking at a series of complications that could result in horrible things, long-term complications, including death. One of the interesting characteristics about COVID-19 is that it's particularly lethal in older folks. So one of the reasons we've had trouble containing the virus is because the younger people tend to have relatively mild illnesses. So if you're 24 years or less, the likelihood of dying from COVID-19 is only 1 in 5,000. If you're between, say, 55 and 65, your likelihood of dying from COVID-19 is about 1 in 50. If you're 85 and older, your likelihood of dying from COVID-19 is 1 in 4. And this is true for all races, for all backgrounds, et cetera. So this is an age-related phenomenon, the way this virus affects people. So again, it's real important, particularly to keep older people and people with pre-existing conditions out of harm's way. And certainly the vaccine strategy is based on that knowledge. Get the people who are most vulnerable the vaccine as quickly as possible.
Well, thank you so much again. Dr. Nabert, maybe in the interest of time, you might want to introduce the next speaker and then we'll take all the questions.
All right, and I think some of the questions in the chat box will actually be answered by Dr. Despani. Dr. Despani is a virologist and has worked actually with coronaviruses in his past and is going to talk with us today about the mutations that are being reported and what that means to us in terms of danger and risk with coronavirus. Dr. Despani?
All right. Can I share the screen? I'm not allowed to share the screen. Either that or I think one of the organizers can upload my slides.
You're co-host now.
Oh. Can everyone see the slides?
Yep.
Okay. Mm-hmm. All right, so everyone, I'm going to be giving my presentation on what I've dubbed Virology 101, which is a corollary to my prior talks, which were Vaccinology 101 and Vaccinology 201. And as mentioned beforehand, it will be on the mutational landscape. And in the interest of time, I'll be moving rather quickly, and it's only about 5 slides. So just to reiterate what has already been mentioned regarding SARS coronavirus 2 and our national landscape, what we're seeing is that we're now up to 20.4 million cases. And that has resulted in 350,186 deaths. And the reality is that that number will probably hike even more before we have gotten the Pfizer and Moderna vaccines into more arms, much less the approval of multiple vaccines. And again, deaths are continuing to rise during this third wave of the pandemic, with fears of a new variant emerging, which you probably heard on the news, both with regards to a new variant coming from the United Kingdom, as well as another from South Africa. And in order to actually understand what a variant is, you also need to understand what is antigenic drift. So antigenic drift is a term that basically refers to an accumulation of what are called point mutations, or individual mutations within the genetic code of a virus that actually alters its amino acid composition. And remember, amino acids are just really these biomolecules that, again, make up proteins. And again, all living things really need amino acids, including yourself. So whenever you have those mutations in the amino acid composition, that can actually influence viral properties. So for instance, you see in this image that you have a virus. This is just a schematic that once it actually undergoes these point mutations, you actually alter the receptors that are available for recognition by the immune system. And that is an example of what you would call an escape mutation, wherein something that was recognized by the immune system can no longer be recognized by the immune system. And what is an example of that would be, for instance, that you have an original viral strain, in this case, the SARS coronavirus mutation that was originally there, which again, I should probably also note that that has also mutated since its first emergence in Wuhan. And what's happening is that you have the, for lack of a better term, the immune system is able to recognize by the existing viral strain via unleashing B cells that provide antibodies, and those antibodies recognize what are called epitopes. Epitopes can be those little red, marks in the diagram. But once you have this mutated strain due to these point mutations that alter the amino acid composition, now you have antibodies that can no longer bind to that same epitope. And then that is what makes a existing vaccine no longer efficacious. So what's going on currently is that we actually have what are two new SARS coronavirus 2 variants. And one of the names, at least for the one in the UK, is B117. And that was actually first identified in the United Kingdom. And actually, there have been over 5,000 sequences of this variant that have been identified. And then there's another variant that's in South Africa, that's B1351. And that one, in patient samples, we have seen more than 300 unique sequences. And what's interesting about both of those variants under investigation is that they both possess this critical N501Y point mutation. And when I mean by the N501Y is, again, we're going back to the importance of amino acids, where N refers to the amino acid known as asparagine. And at the 501st position within the SARS coronavirus 2 genome, that asparagine has actually been mutated to a tyrosine. And that point mutation, interestingly enough, was actually first identified in Italian samples on March 19th of 2020. So what we're suspecting is that there is actually some type of selective pressure that's actually prompting this this N501Y mutation within both the UK and South African, patient samples to actually become the new dominant variant. And again, to understand this, you need to have at least a high level understanding of how natural selection works, right? Which is basically this idea that whenever you're in a certain environment, the mutations in genes that exist in the environment are typically good, but then whenever you are under a pressure of any kind, you typically evolve what are called escape mutations. And those escape mutations can be classified into two types. There are what are called driver mutations and then passenger mutations. So what's going on, at least what we know with regards to the UK variant of the investigation, is that there are 23 total point mutations, and the N501Y is, again, the predominant driver mutations, and then that actually has 22 other mutations that are coming along as passenger mutations. And again, we know that this N501Y is important because it also emerged de novo in South Africa, which again is geographically distinct or circumscribed from the UK patient population. So that N501Y point mutation occurs within the receptor binding domain of the spike protein. And remember, the spike protein is what is utilized in order to bind to the ACE2 receptors on a host cells. So now the question that needs to be asked is, how does this affect our current vaccine landscape? And despite reports of a 70% increase in the infectivity, which again, the infectivity is what we would call in the scientific community R0, which basically is the basic reproduction number that determines how quickly a virus can spread throughout a population. The reality is that the clinical opinion remains adamant that the vaccines will remain effective against this variant, or rather both variants that contain the N501Y mutation. And one reason behind that is that there is also a phenomenon known as cross-reactivity, wherein even though a new variant may not be recognized with 100% efficacy, for lack of a better term, it will still be recognized at least with, say, 40 or 50% or 60% cross-reactivity. So there will still be some effectiveness. It just will not be as it was before. And then, as pointed out so eloquently by Dr. Corin, you also have to consider the fact that the mRNA-based vaccines that have been developed by Moderna as well as Pfizer are actually customizable platform technology. And again, these are technologies that I have actually worked on. Even as Dr. Corin said, he mentioned as well the RNA interference, which is small interfering RNA, and I've worked on that as well. And again, what we're doing is we're moving forward from this old school interpretation of vaccinology, wherein you basically utilize a killed or an activated microorganism and you juice it up with an adjuvant and then you just administer it to the patient. Well, that's very few decades ago, right? Now we're moving into this era where you have, again, a platform technology wherein if you have a new strain or a variant, all that's necessary is you just take the new genome under investigation and you develop a new vaccine candidate around that. So this means that you can now develop vaccines rapidly and immediately get them into being tested in the human body. And in fact, in Moderna's case, the reality is that the SARS coronavirus 2 draft genome was first released on January 11th by Chinese scientists. And within 2 days, Moderna already had a vaccine candidate ready to go. So this is how quickly things move with these mRNA-based vaccine candidates. And then just to reiterate how this even works mechanistically and also logistically, it's just that, okay, so we have a new a new coronavirus that actually has this N501Y mutation. So all you do is you look at the genome, and then from there you sequence a new mRNA that can actually like code for the spike protein that contains any mutations within that spike protein. And again, you run tests on that after injecting it in animal studies and then into humans. And then afterwards, you now have a new vaccine candidate that can actually produce a humoral or antibody-mediated response. So that's all.
Dr. Despani, I thank you for your outstanding contribution. Let me just ask a quick question before we go to the whole group. If I understand your presentation, the new mutations that have been announced both in UK and South Africa,
yes,
don't necessarily pose a significant problem with the immune process started by the current vaccines. Is that correct?
That is correct. So basically, when you have antigenic drift, this can actually become a problem whenever you have like multiple consequential mutations, or aka driver mutations. But as far as we can tell, between the UK and the South African variants under investigation, only the this N501Y seems to be the driver mutation of consequence. And just so you're aware, like for virologists are always looking at all these different variants and whatnot. And before N501Y became the variant to worry about, we actually were paying a lot of attention to D614G. So that was a aspartic acid mutation at the 614 things, either 614 or 644, so, whichever, then it mutated to a glycine. So that was kind of the big predominant strain, not strain, but rather the big predominant variant before this new N501Y became the most important one. But so again, our best expectation is that the vaccines will still be efficacious, but even if they lose some efficacy, it would not be something where it's like, oh, it just went from 95% efficacious down to like 40%, right? Because again, this mutations are an iterative process where again, they're 1-point mutation and 2-point mutations. Now, you know, it gets pretty bad whenever you start to have like 30 to 50 to 100 mutations. So.
Well, thank you again so much. Let's go to the chat box, and I'm going to get questions for all three of our outstanding speakers. I certainly appreciate you, Dr. Hood, you, Dr. Corrin, and you, Dr. Despani, for bringing us this interesting information. Dr. Barbara Stevens.
Okay, we're gonna try to do a rapid fire session to try to get as many questions in as possible. We've got a bunch. I think the first, there have been several questions about the safety of the vaccines in people who have allergies, whether it be to shellfish, eggs, milk, or if they have immune problems, you know, SLE or other immunologic related chronic conditions. What is the safety of the two currently available vaccines for use in these type individuals?
Sure, I can take that. It's a really important question. In the clinical trials, we excluded people who had multiple allergies. So if somebody has multiple allergies, they need to confer with their physician and have a discussion about risk versus benefit. Obviously, everybody's an individual, and you need to know the severity of those allergies, the type of allergies, and also your susceptibility to complications of COVID before you make that decision. But to be clear, those people were excluded from the clinical trials. Also, people who have immunocompromised status were excluded from the clinical trials. So people on high doses of steroids or other agents, anti-TNF alpha drugs were also excluded from the trial. So again, for those folks, there has to be a conversation with individual physicians who need to make that decision with the patients.
Let me ask you a question in there, Dr. Corrin. Knowing that we're not giving direct advice to our patients, but if you had a patient with SLE or some of these allergies and problems that Dr. Neighbor Stevens spoke about, how would you start that conversation?
Yeah, and that's reasonable. So if somebody had stable SLE and wasn't on particularly high doses of steroids, I would probably say go ahead and get it, because SLE potentially could also make you more vulnerable to complications of the virus. So you have to make that trade-off. If somebody was on super high doses of immunosuppressive agents, I'd be more reluctant. Or if somebody has had horrible problems with flu vaccine, I probably would stay clear. There's also been some reports of people that are sensitive to propylene glycol having reactions. Propylene glycol is a stabilizer agent that's used in the vaccine. It's used in a lot of other drugs and cosmetics and other things, and it usually does not cause any problems, but there's a very small fraction of people that could have a reaction to that. So if you have a documented history of that, that might be a person I would avoid using the vaccine in.
Thank you. Dr. Neighbor?
Okay, and now I'm going to tie a couple of questions together. Earlier, there was a question about the etiology of the virus when it first arrived in the United States. The question was specifically, did it come from Europe or did it come from Wuhan? But I want to tie that question in because I think it's related. And I suppose this goes to Dr. Despaigne. The second part of this question is, does the N501Y variant equate to the G614D variant? And to clarify the difference. And then the third part of that question was, are these variants occurring in places where there is a lack of testing?
And can we do brief answers so we can get as many as possible?
Yeah. So first off, is it occurring in places where there's a lack of testing? Yes. To the best of my knowledge, it's already the UK one has already spread at least to 10 plus countries, and the South African one, I believe, has spread to at least 4 countries. And I doubt that, I know one of the countries that the UK strand was first recently identified in was the Philippines. So I don't think that the Philippines can move as fast on testing. So yeah, it's obviously spreading. The reason why it's spreading is, again, because this mutation is proving to outcompete its cousins, for lack of a better term. So it's just more effective. To answer the question regarding does N501Y equate to its D614G, no, but they have the same net effect. Both mutations actually increase the affinity or the binding tightness of the SARS coronavirus 2 spike protein to the ACE receptor. So basically it's trying to evolve in such a way that once it binds, it will take less variants to initiate an infection. So typically it's always been that it takes about 1,000 variants to initiate an infection with SARS coronavirus 2. But again, the tighter it binds, the less variants it needs to cause a problem. And just so you know, a variant is just an individual virus particle. Yeah.
Thank you very much.
And the next one is to Dr. Corin, and I think maybe you might need to clarify something. So the question is, should the US have waited for the AstraZeneca vaccine so that our bodies are self-reliant viral killer machines?
No, I think that the messenger RNA technology is superior in my opinion, and we have that through the Pfizer and the Moderna products. The advantage of the AstraZeneca product is that it has easier storage capabilities and easier logistics. So I don't think we should wait for it. And people ask me all the time, which one should I take? And I say, whichever one you can get first between Moderna and Pfizer. And we're also working on Novavax, which is another type of technology that doesn't involve injecting a virus. You just inject genetically engineered spike protein with an adjuvant. And I think that would be superior also. The problem with the other type of vectors is that you're more likely to create antibodies that are less specific. I'll also just give people a little bit more information who are nervous about these new variants of the virus. There's a couple of things you should know. both the Pfizer and the Moderna products are actually more than one, create more than one antibody. So the Pfizer product actually has what we call 5 valences, which means that there's 5 different parts of the spike protein that are being programmed to develop antibodies against. So even if there was one segment that became a problem with a variation or mutation, you have all these other particles that are available to make sure the spike protein doesn't interact with the human receptor. The Moderna product has 2, and the Moderna product also has a longer sequence that covers more area of the spike protein, if you will, so that even if there's one area that doesn't quite match anymore, the other areas should match. So I think I have a low level of concern that these vaccines will stop working. The other thing that's really important, I just read this, and Jordan might want to comment on as well, is that people that are getting infected with the new variants are not people who got the previous variants. So if this was kind of a new virus and put people at risk, people who already had COVID-19 would get reinfected. But in fact, the people that are getting the new variant are people who have never been infected before. And that bodes extremely well for the continued efficacy of the vaccine.
Thank you again, Dr. Neighbor.
All right, there is a question. Would one of you comment, is there any data on the effect of mRNA vaccines on spermatogenesis or fetal development?
Yeah, I would say that that's way too early to deduce that because again, right now we've only gotten to emergency use authorization. There has not been any official like, okay, now this can be in all sorts of, well, again, it needs to get through to phase 4, basically. So emergency use authorization is not the same thing as phase 4. Phase 4 is basically that it's been approved by the FDA, but even though a drug is approved by the FDA, there's still ongoing safety and, testing in the population, even after approval. So just so you know, like even after a drug is approved, it's still being monitored for years on end. So.
My comment.
We have one more question that I really want to get in, Dr. Maxey.
My comment on spermatogenesis, if you're dead, there's no spermatogenesis. Go ahead.
So this is to any of the presenters. What happens if the second shot does not come on time? Whether it's 21 days or 28 days, whichever one.
Do you want to jump in, Dr. Hood? Or you want me to answer that?
You go right ahead.
Sure. So the protocols were set up so that for Pfizer, the second dose was given around 21 days later, and for Moderna was 28 days later. But the truth is that we don't know if that second dose is absolutely necessary. We think probably it will sustain the duration of time in which your antibodies will be produced and the time in which you will not be susceptible to COVID-19, but whether or not it's absolutely essential, we don't know. There haven't been some public health figures that have stated, well, maybe we should not be giving the second dose to anybody until everybody gets a first dose, because this timing of 21 days and 28 days is somewhat arbitrary. But as I showed you from that Kaplan-Meier curve, or the separation of the curves, is literally 10 days after the first dose, we're finding that people who are vaccinated have extraordinarily low risk. So the second dose may or may not be required, and we don't know if the timing is really essential. That was just a guess that was made by the manufacturers, and that's what we did in the studies. And I'll also just mention that pregnant women were not involved in the studies, although there are studies ongoing now looking at the vaccines from both Moderna and Pfizer in pregnant women. And I'm not familiar with any specific data on spermatogenesis.
Another question
So
that
if I could just add to that, the new virus, the AstraZeneca one, has not been approved here. When you really read the data there, it actually suggested that in a small subgroup, spreading out the dose to longer actually get more efficacious. So instead of the usual month, if they spread it out 8-12 weeks, there seemed to be a stronger efficacy. So I think what Dr. Curran said is correct. 2 doses, 3-4 weeks apart is kind of arbitrary, but right now that's what the FDA recommended here. And I know there's discussion about maybe us considering that, but I don't know anybody that's recommending.
Dr. Hood, could you speak to making sure you get the second dose from the same first dose, or does that really make a difference?
I think that's critically important. The recommendation is that your second dose should be the same as your first dose, because we don't have any data about changing the doses, whether it's messenger, especially with the adenovirus, I'm not really sure. So yes, I think it's critically important right now with the data we have that it be the same manufacturer.
And then we have two questions related to what happens after you get the vaccine. The first is, once you're vaccinated, can you still be a COVID virus carrier? And the second one is, if you had COVID, should you get the vaccine?
Yeah, you can actually still spread the virus after you have the vaccine. So the virus will, the vaccine will protect you from any severe systemic effects of the virus. But obviously, if you're vaccinated and somebody with COVID-19 coughs in your face and you touch your hands and then you take the secretions from your hands and touch somebody else, you can pass that along. So people who've been vaccinated still need to take precautions. Hygienic measures are still important for people that have been vaccinated. As far as people who have gotten COVID already, we actually found that between 2 and 3% of the people who were in the studies for both Moderna and Pfizer already had COVID. So when people came into the study, we did antibody testing, but that didn't influence whether or not we put them in the study. And for people who already had COVID or been exposed to that, we still saw a difference in people who were vaccinated versus people who weren't. And that may be related to the persistence of the virus or other factors. So you can think of the vaccine in somebody that's had COVID already as the booster. I've had a number of patients already approach me about that. They've had COVID, should they get the vaccine? And I've advised them, and this is kind of off-label information, so take it as you will, just my opinion, that if I had COVID in the past, I would only take 1 dose of the vaccine, but I would still recommend it.
I'd like to ask, go ahead, Dr. Hood.
If I could add to that, when you first get the first dose, there's a 7-10-day period that you're still vulnerable. So there have been reports of folks getting the first dose and actually themselves coming down with COVID, although I don't think it was severe. So it does take 7-10 days to get some type of immunity response where you are still vulnerable. And then the second dose is the dose that takes you to 94%. So I think the other, I think the other question was.
Dr. Hood.
I have a Facebook question that just came in. Can you kill the virus, or is it only made dormant by drugs?
A virus is not alive. A virus is like neither alive nor dead. So it's like viruses they're just, biochemistry. So all you're doing is you're just inactivating it. But one thing that's very important is that people do need to focus on washing their hands, because one thing that people often forget or might not even know is that coronaviruses are enveloped viruses, meaning that whenever they actually come out of infected cell, they steal parts of the plasma membrane, which is like the fat that makes your cell, and they use that for their own structural integrity. So actually, if you use soap or like detergents and whatnot, that actually disrupts that the lipid membrane that it uses for the envelope and destroys it. So it's actually easier to destroy coronaviruses than it would be for a non-enveloped virus, like for instance, an adenovirus. So yeah, so wash your hands.
Dr. Hood, I want to ask you a question. As past president of the National Medical Association, would you give our audience public health advice on how they should conduct themselves even after the coronavirus in terms of cleanliness and spacing, et cetera?
Yeah, so I think this message needs to go out. We're talking about vaccines, and we're all stating that everybody should get vaccinated. But we also realize, we don't have the best public health system systematically in this country. It's very fragmented and dependent upon the state and locality. So even that the vaccine is being rolled out, it's going to take a while for the population to kind of get vaccinated. On top of that, we've got subpopulations that, no matter what message we give, are going to be reluctant to get the vaccine. So here's the message for everybody, no matter who you are. I think the message is you need to do safe mitigation. You need to wash your hands, wear a mask, social distancing, stay out of crowded environments. I think what we don't talk enough, there are natural ways we can boost our immune system. I don't think they're as effective as a vaccine, but that means relaxation, plenty of sleep. I believe in vitamin D, C, zinc, and trying to eat healthy and exercising. I think really we're looking at 70, 80% herd immunity. And so even if we don't get to everybody, it's important that everybody who may not take the vaccine continue to do the safe and wise thing. Whether you take the vaccine or not, as stated by Dr. Corin, you still need to practice that.
Yeah,
Well,
and,
thank you so much.
and Dr. Maxey, can
yeah,
I?
go ahead.
Yeah, so I see that there's a question where someone asks, why can't a vaccine be made that generally attacks, certain base components of a virus like nucleic acid, protein capsid, lipid envelope? That actually is what these vaccines do. Like they're basically leveraging like an artificial, for lack of a better term, version of the spike protein so that it can draw your immune system to recognize and attack that like the spike protein, and the spike protein is found on the capsid. So think of the immune system or the antibody response as like that's an indirect way. The direct way to actually treat viruses are, again, it sounds simple, but that's the name, antiviral drugs. And to the best of my knowledge, there currently is a drug that it was recently tested in ferrets. Ferrets are often used as an animal model for respiratory diseases. And it was found to be very efficacious against SARS coronavirus 2. And now they're moving it into preliminary, studies in humans. And basically the way that acts is that it acts as it interferes with the coronavirus's RNA-dependent RNA polymerase. So RNA-dependent RNA polymerase is basically what the virus uses to make its nucleic acids. So this drug just basically like shoves into the RNA-dependent RNA polymerase and makes it stop spitting out more nucleic acids. So there are actual drugs in the pipeline, and these are actually what would be called pre-exposure prophylaxis, which basically means that you take it and you would hopefully not get sick at all. But again, that's at the very early stages of clinical studies.
Thank you so much. Just want to speak briefly about some of our upcoming programs. Dr. Keith Norris, who is one of the world's experts on vitamin D, is putting together one of our next programs on the efficacy of vitamin D and why it's good and any risk with that. Dr. Richard Williams is going to be presenting a program later in first quarter. We have some pastors who are also physicians and scientists who are going to speak on the 10th of January on avoiding COVID, and a number of other programs on maternal fetal mortality and some other exciting programs. So we invite you to keep up with us. I want to honor and thank each of our speakers, Dr. Hood, Dr. Corin, and Dr. Despani, and my co-moderator, Dr. Barbara Stevens. And I'd like to remind you that we are not giving direct medical information here, but rather information that you can take to your physician or provider. And I also remind you to look on the website of blackhealthtrust.org. Everything is archived on the previous presentation. And if you're a layperson and you need consultation from an expert physician, look at biometrics.secondopinionexpert.com. Dr. Neighbor-Stevens, any words?
I think that we are caught up with the chat, and we are going to need to stay vigilant in terms of our own health as well as our community and what's happening around us.
I want to say two things, and I'm going to ask the chair of our vaccine task force of the Black Health Trust to speak. I want to honor our president-elect, Dr. Villanueva, with Dr. Walks, and followed by our takeout by Brenda Leager. Dr. Walks, briefly.
There we go. Good afternoon, everyone. I would just really like to thank all of the presenters that we had today. Dr. Hood, it's been many, many years since I saw you in San Diego. Invite me back anytime. Happy to come to San Diego. Dr. Maxey, what you all do here is just fantastic. The level of science provided with this level of clarity just makes it useful. And I'm happy to be a part of it. And I hope that we can continue to do this kind of good work. So thank you very much.
Thank you very much. Ms. Brenda Leager? Shion, I saw her earlier.
Can you hear me now?
Yes, we can.
Oh, wonderful. So I just want to say, I've got my tissue here because I've been crying. You wonderful physicians, when you were speaking your name and saying what you do and who you are, I got so full because as a child of a farm in Alabama with dreams, you have lived your dreams and you are supporting and helping other people. And I just want to say, we're so blessed to have you, and thank you, Dr. Maxey. Thank you, each and every one of you. And I just know that as we raise our awareness, we raise the vibration of this world to be better, and this too shall pass. Because I believe, I believe, I believe, oh, I do believe. People get ready, there's a change coming. Don't need no baggage, you just get on board. All you need is faith and understanding and raising that consciousness to what these people are telling us. You don't need no ticket, you just get on. You just get on board. And with that, I'd like to say thank you, God, for these lives that we're all one, living in this great, great universe. And we're here to love and help one another. And we continue to be that which we came to be. And so it is, and so let us be. And so I allow it to be. And thank you so much.
Thanks, everybody. Have a great new year, a great rest of the day.
Bye.
Bye.